Hello everyone and welcome to Roadmap. So today we are going to learn about the stomatic embryogenesis and the applications of micropropagation. What exactly is stomatic embryogenesis? Now you must have heard about stomatic embryogenesis or it must be in your syllabus. So stomatic embryogenesis is the process of regeneration of embryos from stomatic cells, tissues or organs. This process is called as the stomatic embryogenesis. Now stomatic embryogenesis may result into non-zygotic, that is stomatic embryos which are directly from the stomatic organs, or the pathogenic organs which are formed from the unfertilized egg, and the androgenic embryos which are formed from the male gametophyte. So when the term stomatic embryo is used, it generally implies that it is formed from the stomatic tissues and that are formed under certain in vitro conditions. So there are certain environmental conditions that are used for the formation of the stomatic embryogenesis. So the stomatic embryogenesis, they are structurally similar to the zygotic embryos and they can be excised from the parent tissues, and they can induce it to germinate in tissue culture media. So the development of the stomatic embryos that can be done in plant cultures using the stomatic cells. Now, when we say that the development of stomatic embryos can be done in plant culture using stomatic cells, then the question must be arising to you that which cells are used. So basically the cells which are used are the epidermis, secondary root phloem and the parenchymatous cell. So stomatic cells that are arise from the cluster of meristematic cells in the callus or in the cell suspension. So stomatic embryos, they arise from the single cells which are located within the cluster of meristematic cells in the callus or the cell suspension. So first what happens, the pro-embryo is formed first, then it develops into the embryo, and then finally a plant is formed. So how the stomatic embryogenesis happens, that the stomatic embryo is formed from the stomatic tissues or organs. Then first what will happen, the pro-embryo will be formed first, then the formation of the embryo will take place, and finally it will form the plant. Now, there are two types of stomatic embryogenesis. One is the direct embryogenesis and the other is the indirect stomatic embryogenesis. Now, it is same like the organogenesis. In organogenesis also there is direct organogenesis and indirect organogenesis. So here in direct stomatic embryogenesis, what it is exactly, when a stomatic embryo is formed, it develops directly on the excised plant, that is the X plant, and it does not undergo the callus formation. So it is called as the stomatic embryogenesis. So direct stomatic embryogenesis, it means that the stomatic embryo is formed without undergoing the callus formation. And indirect stomatic embryogenesis, you must have got now that it is with the formation of the callus or from the cell suspension culture, the indirect stomatic embryogenesis takes place. So coming to the direct stomatic embryogenesis, it is formed due to the presence of the pre-embryonic determined cells, that is PEDC, which are found in the certain tissues of the plant. Now, the direct stomatic embryogenesis avoid the introduction of somaclonal variation within the propagated plants. So this is the characteristic feature of the direct stomatic embryogenesis. Now, this is the basic of direct embryogenesis, that it does not undergo callus formation. It is formed due to the presence of the pre-embryonic determined cells which are found in certain tissues, and it has got the characteristic feature that it avoids the introduction of somaclonal variation in the propagated plants. Now coming to the indirect stomatic embryogenesis. Indirect stomatic embryogenesis, as I said, that the explant is undergoes the callus formation, from which the cell suspension culture can be raised. And there are certain cells, that is the induced embryonic determined cells, or IEDC, from the cell suspension, that can form the stomatic embryo. So the embryogenesis is made possible due to the presence of the growth regulators that make stomatic embryogenesis, and that are formed under the certain chemical environmental conditions. So there are certain environmental conditions, and it is possible with the help of the growth regulator. So this is about the indirect stomatic embryogenesis. Now, for the better explanation, from the figure you can see that from a plant, explant is taken, that is leaf, then it undergoes the formation of stomatic embryo directly without undergoing the callus formation. Then it undergoes formation of the shoots and roots, and then that plantlet is taken into the green environment for the formation of plant. Where this is the direct embryogenesis, stomatic embryogenesis. Now the indirect stomatic embryogenesis, what it is exactly? The plant forms the from the plant, explant is taken, that is leaf, then it undergoes the callus formation, or it will go the cell suspension culture formation. Then cell suspension culture formation will form the callus formation. So ultimately callus formation is going to take place. Then that plantlet undergoes the formation of shoots and roots, and then it is that plantlet is taken into the green environment for the formation of plant. So this is the indirect stomatic embryogenesis. Now, the media that is used for the stomatic embryogenesis is white media, or the MS media is used, and there is the addition of the amino acid, which is the L-glutamine, which promotes the embryogenesis. Then the oxime, called as T4-dichlorophenoxyacetic acid, is used for the embryo initiation. And on low oxime or no oxime medium, embryogenic clump develops that matures in that forms the mature embryos. Now the indirect stomatic embryogenesis is commercially very attractive. Large number of embryos can be generated from the small volume of the culture medium. So the stomatic embryos formed has got the good regeneration capability, and they are also synchronous. So this is about the basic of the stomatic embryogenesis. Now, the artificial seeds or the encapsulated seeds can be formed from the stomatic embryos. Now, artificial seeds can be made by encapsulation of stomatic embryos. The embryos are first coated with sodium alginate and then the nutrient solution, and then further they are dipped into the calcium chloride solution. So the calcium ions, what they do, they induce the rapid cross-linking of sodium alginate to produce the small gel beads, each containing the encapsulated embryo. So these artificial seeds or the encapsulated seeds, can be used, and they can be maintained in a viable state till they are planted. So this is about the artificial seeds from the stomatic embryos. Now, coming to the application of micropropagation, micropropagation has become the suitable alternative method, and it is a conventional method of vegetative propagation of plants, and it has got the certain advantages. The first is like the high rate of plant propagation. Now, through micropropagation, a large number of plants can be grown from a piece of plant, that is explant tissue, within a short period of time. Then micropropagation can be carried out anytime throughout the year, irrespective of the seasons. So many plants that are highly resistant to conventional propagation, micropropagation is the suitable method. Now, the small sized propagules obtained in micropropagation can be easily stored for many years, that is the germplasm storage, and it can be transported across the international boundaries. So this is the first application of micropropagation. The second one is the production of disease-free plants. Now it is possible to produce disease-free plants through micropropagation. For this, the meristem tip culture are generally employed to develop the pathogen-free plants. And micropropagation is successfully used for the production of virus-free plants of sweet potato, yam, and cassava. So this is the second, that is the production of disease-free plant application. The third is the production of the seeds in some crops. The micropropagation through axillary bird proliferation method. It is suitable for seed production in some plants. Now this is required in certain plants where the limitation for seed production is high and the degree of genetic conservation, like the examples such as cauliflower and the onion. This is the third one. The fourth one is that it is cost effective process, that the micropropagation, it requires less space, and many, like millions of plant species can be maintained inside the culture vials in a small room or in a nursery, and it is a cost effective process. The production is relatively low. It requires low cost in many developing countries such as India, where the manpower and labor charges are low. So it is a cost effective process. Then the next one is that the automated micropropagation. Now it is also possible to automate micropropagation at various stages. Bioreactors have been set up at large scale for the multiplication of shoots and bulbs. So workers employ robots in place of laborers. We can use robots for micropropagation and further reduces production cost of the plants. So these are the application of micropropagation. Now there are certain disadvantages of micropropagation. The first one is the contamination of culture. Now during the course of micropropagation, several slow-growing microorganisms contaminate and grow in cultures like the bacillus species. The microbial infection that can be they can be controlled by addition of antibiotics or fungicides. Now this will adversely influence the propagation of plants. Then the brewing of medium, micropropagation of certain plants is associated with the accumulation of growth inhibitory substances in medium. Now these substances are the phenolic compounds and they can turn the medium into dark color. So the phenolic compounds, we know that they are toxic and they inhibit the growth of tissues. So brewing of medium can be prevented by the addition of the ascorbic or the acetic acid to the medium. So this is the second disadvantage of the micropropagation, that brewing of medium. The third is the genetic variability. When micropropagation is carried out through the different cultures such as the shoot tip culture, genetic variability is very low. Now, the use of adventitious shoot is associated with the pronounced genetic variability. Then the vitrification during the course of repeated in vitro shoot multiplication, culture exhibit water-soaked or almost translucent leaves. So shoots cannot grow in such condition and many a times they die. Now this is nothing but vitrification. Now vitrification can be prevented by the increasing of the agar concentration, that might be from 0.6 to 1% in the medium, and that can be increased agar concentration. It reduces the growth of the tissues. Now the cost factor. Now micropropagation technique has got the expensive equipment and sophisticated facilities, and the trained manpower is needed, so it limits the use of the micropropagation. Now, production of disease-free plants. Now, many plant species are infected with pathogens such as viruses, bacteria, fungi, and nematodes that cause the systemic diseases. Now, these diseases do not always result in death of plants. Now, what they do is they affect the quality and the yield of plants. Now, the plants that are infected with bacteria and fungi respond to the chemical treatment by the bactericides and the fungicides. Now, it is very difficult to cure the virus infected plant. Viral diseases are easily transferred in seed propagated as well as the vegetatively propagated plant species. And the plant breeders are always interested to develop the disease free plants, like they have become a reality through the tissue cultures that disease free plants can be produced. apical meristem with low concentration of viruses. Now, the apical meristem of pathogen infected and disease harboring plants, they are either free or carry a low concentration of viruses. Now, that is because of the few reasons that it happens. Now, the first one is that the absence of vascular tissue in meristem through viruses readily move in the plant body. Then the rapidly dividing of the meristematic cell, the continuous dividing of meristematic cells with high metabolic activity, they do not allow viruses to multiply. And the virus replication is inhibited by a high concentration of endogenous oxygen and shoot apices. Now, the tissue culture technique employing meristematic tips is the successful method which is used for the production of disease-free plants that are caused by certain pathogens like the viruses, bacteria, and fungi. Then third one is methods to eliminate viruses in plants. Now plants are infected with many of the viruses, and the nature of some of them is completely unknown. The usage virus-free plant implies that the given plant is free from all the viruses, although it may not be always true. So the methods that are commonly used for the elimination of plants like the heat treatment of plant, meristem tip culture, chemical treatment of media, other in vitro methods. So we'll learn one by one about them. Now, coming to the heat treatment of plants or the thermotherapy, what happens is in the early days before the advent of meristem culture, in vivo eradication of viruses from plants has to be achieved by heat treatment of whole plants. The underlying principle is that many of the viruses in plant tissues are either partially or completely inactivated at higher temperature with the minimal injury to the host plants. Now, this thermotherapy, the temperature is 35 to 40 degrees Celsius and it is carried out using hot water or hot air for the elimination of viruses from growing shoots and buds. Now, there are two limitations for this therapy. Like most of the viruses are not sensitive to heat treatment, and many of the plant species do not survive after thermotherapy. So, these are the two disadvantages of the heat treatment, and it has not become the popular therapy. Now, the meristem tip culture. For the viral elimination, the size of the meristem used in culture is very critical, and it is because of the fact that the establishment of the gradient in plant tissue. The regeneration of virus-free plants through culture is inversely proportional to the size of the meristem used. So, meristem tip explant used for viral elimination is very small, and stereoscopic microscope is generally used for this purpose. Now, the meristem culture is influenced by the factors such as the physiological condition of the explant, that actively growing birds are more effective than the thermotherapy, which is prior to the meristem tip culture for the certain plants. Heat treatment is given first, and then the meristems are isolated and culture. Then the culture medium used, like the MS medium, with the low concentration of oxygen and cytokinin ratio is ideal one. Then coming to the chemical treatment of media. Now there are some workers have attempted to eradicate the viruses from infected plants by chemical treatment of tissue culture media. Now the commonly used chemical growth substance for this purpose was cytokinin and antimetabolites. So antimetabolites such as the thiouracil and the acetyl salicylic acid. So the reports that came from the element from this uh, experiment was that the elimination viruses by chemical treatment of the media, addition of cytokinin suppresses the multiplication of certain viruses, while for some other viruses it actually stimulated. So few of the viruses they will suppress, and few of the viruses they will get stimulated by the chemical treatment of the media. Now, there are certain other in vitro methods also that I use, like the besides the meristem tip culture or thermotherapy or chemical treatment of media. That is, the callus cultures have been successful. They are derived from the infected tissue and they do not carry the pathogens throughout the cells. And there is uneven distribution of the tobacco mosaic virus in tobacco leaves that was exploited to develop the virus-free plants of tobacco. Now, stomatic cell hybridization or gene transformation and somaclonal variations are also used for disease-free plants. Now, if you have to learn what are somaclonal variations and their applications, I have already uh, uploaded a video and that so that you can get that concept and you can have a better understanding from that. So there are other few methods such as the elimination of pathogens other than viruses. Now, besides the elimination of viruses, meristem tip culture and callus culture are also used for the eradication of bacteria, fungi, and mycoplasma. Now, the examples such as the fungus Fusarium roseum that has been successfully eliminated through the meristem cultures from the carnation of plants. There are certain bacteria that can eradicate from the carnation plants by using meristem culture. So these are few examples of pathogen elimination other than viruses. Now, there are certain merits and demerits of disease-free plants. Now, the culture techniques like the meristem tip culture is the most reliable method for the virus and the pathogen elimination. But it requires good knowledge of the plant pathology and the tissue culture. So, the virus-free plant exhibit the increased growth and vigor of plants like higher yield, increased flower size, and the improved rooting on of stem cuttings. So the virus-free plants they are most susceptible to the same virus when exposed again. Now this is the major limitation. Reinfection of disease-free plants can be minimized with the good knowledge of the greenhouse maintenance. So these are few merits and demerits of disease-free plant production. Now here we complete the lesson of the somatic embryogenesis and the applications of micropropagation. Now from in the further video we'll learn about the embryo culture and then the applications of embryo culture. Now if you like the video, don't forget to click on the subscribe button below and share video and keep watching. Thank you for watching.